Welcome to lecture 18 of Bio 120. Today we're going to be looking at different ways in which bacterial cells regulate their gene expression. So we're going to start our exploration by looking at some of the principles of gene regulation. So the process of gene regulation, it's imperative for microorganisms that are living under rapidly changing conditions. Processes that are no longer needed get shut off. Processes that are needed will only be turned on when they're needed. So they have to be initiated. So microorganisms have developed efficient ways to maintain a tight control over gene regulation and gene expression. For example, think about the downregulation of the lactose enzymes when lactose is absent. We're going to be looking at that in more detail when we think about it later today. So there are two different levels in which we can have gene regulation happening. At the DNA level, we have the area of the promoter that is going to be the site where the RNA polymerase binds. And that promoter could have areas of activation and areas of repression. After the transcript has been made and we have now messenger RNA, we can have also levels of regulation at the RNA level, especially because the RNA has 5' untranslated regions as well as 3' untranslated region. At the end, we can make a protein out of that mRNA, and that protein can be controlled by different enzymatic mechanisms, including, for example, feedback inhibition, protein-protein interactions, degradation, as well as covalent modifications. So, for example, let's illustrate one of the ways in which proteins can be controlled by post-translational regulation. And we can think, for example, of a biochemical pathway where we have a starting substrate shown over here at the upper left, and that starting substrate is going to be catalyzed by enzyme A into intermediate 1. After that intermediate 1 will be the substrate of enzyme B that produces intermediate 2. We can have now intermediate 2 be the substrate for enzyme C, producing product intermediate 3, and then intermediate 3 will be the product of enzyme D, producing this end product shown over here. Now, enzyme A is an allosteric enzyme, an enzyme that is going to change conformations when the product that is made at the end of the reaction can now inhibit it, and that is illustrated here in the picture to the right. So for example, here we have your enzyme A, and it has two binding sites. One binding site that we call the allosteric site, where the end product can bind to, and an active site where the substrate can bind to. So in the absence of the end product, the substrate can bind to it and the enzyme is functional. But when the end product is available, the end product can bind to the allosteric site, inhibiting and changing the conformation of the active sites and therefore stopping the pathway. So that's one of the ways that we can look at regulation. That will be the feedback inhibition by allosteric means. But also a protein could be degraded. You can have different modifications like phosphorylation. Here in the bottom right, I have an example of that where we're going to be looking at a response regulator, for example, that is going to be phosphorylated by a kinase. And that kinase now is able to put a phosphate on that response regulator. That response regulator now is on, it is active, it can perform a function, but eventually when the phosphate is removed, that inhibits the function of that response regulator. And then at the end, the last one we can think about are proteolytic processing, where a protein is cleaved by proteases to remove an inhibitory site and then allow a functional unit to work. For example, the DNA gyrase alpha or A subunit has an intane, which is the equivalent of an intron in a protein. So only after degradation and removal of this intane do we have a functional enzyme. So those are some of the ways in which the activity of proteins could be regulated after they have been translated. But we're going to think about transcriptional control. control that happens at the level of transcriptions. And what we're going to be looking at are proteins that are going to bind to regulatory sequences in the DNA. So we're going to have DNA binding proteins, and those DNA binding proteins are transcription factors that can bind and upregulate or downregulate transcription of a protein. So we're going to see that those transcription factors are going to have sequence-specific loci where they can bind directly. And they're usually binding on the major groove of DNA. So here's an example of that. In this image, we have a transcription factor has dimerized because oftentimes they need to dimerize in order for them to be able to be effective. They have a dimeric domain that allows protein-to-protein contacts that holds the protein dimer together, and at the other side, they have a DNA binding domain that can bind to the major groove or along the sugar phosphate backbone. So here at the bottom, we have an example of a DNA sequence that illustrates the inverted repeats that are recognized by transcription factors. And these are shown over here by this blue and this pink arrow. So notice that the repeats are identical in both directions and therefore are able to be recognized by the transcription factor to be able to bring it and effectually do its job that it needs to do. And as I mentioned to you early, some transcription factors are going to bind to the major groove of DNA. As you know, the major groove is this area here where the helix are separated at its maximum. And here you can see the DNA molecule as a field model bound by a transcription factor who has been dimerized. And that can be illustrated here by this image where a plasmid has been now added to a solution that has transcription factors, and you can see them binding in the DNA molecule. And that ends our introduction to the principles of gene regulation. In the next section, we're going to take a more in-depth look at different ways in which transcription is regulated.